The Baja Biocomplexity project is collecting data using a variety of field techniques and disciplines. The ecological field work takes place underwater. To do this, our team of trained scientific scuba divers must set up underwater instruments, conduct visual surveys, mark animals, collect samples, and execute experiments underwater. It is usually a major undertaking, and we're thankful for the help of the fishing cooperatives we work with. In the morning, we pack up our equipment, load the pangas, and hit the water. We carry over a dozen tanks, dive gear, oxygen and first aid kits, and a variety of field gear like transect tape, measuring chains, PVC quadrats, tissue samplers, data sheets, slates, collecting bags, GPS, and of course, cookies. Our captain charts the course, and our 2 floating research laboratories head out to sea, ready for our underwater adventures. We travel up to 10 miles along rugged desert coastline. We have 9 study sites, each containing shallow and deep depth ranges at reefs that vary in their productivity. At each site, we conduct a variety of visual community surveys of species diversity, as well as experiments with the animals we find. Once at the site, the dive team makes the final preparations and takes instructions from the lead diver.
Remember to stay with your partner at all times, complete a 3-minute safety stop, and exit the water with over 500 psi left in your tank.
We strap on our BCs, check our air pressure, and tighten the buckles on our weight belts. Each tank gives us over an hour at these depths. There's a lot of gear to bring down with us, and we don't want to forget anything at the surface that we need down below.
I feel strong, I feel ready.
Once in the water, we descend to the reef and find the best place to start our work. Sometimes it takes a while to find the same places where we dove several months before. To study annual movements of sea cucumbers, we mark as many individuals as we can with colors corresponding to different depth ranges. When the fishery begins a few months later, we will track whether cucumbers have moved across these depths. To sample the diversity and species composition of the seafloor at each of our study sites, we conduct visual transects 10 to 30 meters long. Along each transect, we identify, count, and measure kelp, corals, fish, as well as invertebrates like lobsters, abalone, snails, and sea cucumbers. We then characterize the bottom type by measuring the percent cover inside randomly placed quadrats. With this information, we can construct models of the nearshore marine food web to look at how various species interact with each other, and also to examine the effects of fishing on the ocean ecosystem. At several sites, we elect a group of settlement tiles to look at recruitment rates of algae and sessile invertebrates. We were surprised how many things we found making homes on the tiles after only a few months underwater. After completing a dive, the team helps each other back on board and the real work begins. Recently, wavy turban snails have become a major target species for the fishing cooperatives. We are looking at growth rates of these snails at several of our study sites in an intensive mark recapture study. Some days we spend hours scraping hundreds of snails to look for tags we put on months ago. When the work is done, we head back to shore to clean off our gear, rush live animals to the laboratory, and meet up at La Preciosa restaurant for some fish tacos.